This method can help answer some key questions in various fields of research, such as detection of precancerous clones, prognostic assessment of leukemia patients following treatment, and identifications of potentially pathogenic mutations in bone marrow donors. Demonstrating this procedure with me will be Spencer. He is a technician in the lab.
This video describes how to combine the error-corrected DNA sequencing protocol with Illumina chemistry and commercially available gene panels to detect clonal single nucleotide variants and small indels at a 1 in 10,000 sensitivity. To incorporate the required unique molecular identifiers, customized 16N-I5 and I7 adapters, use PCR. First, prepare the Q5 master mix, which uses a polymerase with higher fidelity than the commercially available alternative. Then, for each reaction, combine 37.5 microliters of Q5 master mix, 6 microliters of 5 micromolar 16N-I5 adapters, which are the unique molecular identifiers, and 6 microliters of I7 adapters. If the goal is multiplexing, then use different I7 adapters in separate samples. Now, run the PCR using the following parameters: 30 seconds at 98 degrees Celsius, followed by 4 to 6 cycles of 10 seconds at 98 degrees Celsius, 30 seconds at 66 degrees Celsius, and 30 seconds at 72 degrees Celsius. Finish the reaction with a 2-minute extension at 72 degrees Celsius and hold at 4 degrees.